What if I told you that right now, as we speak, there are children walking around who are the first humans in history to have been cured of diseases that have killed millions for thousands of years? Not treated, not managed, completely cured. Using a technology so precise, it can edit individual letters in the 3.2 billion letter book that is your DNA. And what if I told you that this same technology could eliminate hereditary diseases forever, bring back extinct species, end world hunger, and potentially make us immortal? This isn't science fiction. This is CRISPR, and it's already changing everything. Welcome back to Mind and Marvel, where we explore the scientific breakthroughs that are literally rewriting reality. I'm your host, and today we're diving deep into the most revolutionary technology in human history, CRISPR gene editing. Now, I know what some of you might be thinking. Gene editing? That sounds scary. Isn't that how we get supervillains and genetic monsters? And I get it. The idea of editing the fundamental code of life can seem terrifying. But here's the thing. CRISPR isn't some theoretical future technology. It's here. It's real. And it's already saving lives. Just last year, the first CRISPR-based medicine, called Casgevy, was approved for treating sickle cell disease and beta thalassemia. We now have patients who are dying from genetic blood disorders who are completely cured. Not improved, not stabilized, cured. Their bodies are now producing healthy blood cells for the first time in their lives. But that's just the beginning. In the past year alone, researchers have used CRISPR to restore vision to blind patients, grow organs for transplant, create crops that can survive climate change, and even bring back genetic material from extinct species. We're living through a revolution that makes the invention of antibiotics look like a warm-up act. But here's what makes this story even more amazing. CRISPR was discovered completely by accident. Scientists studying how bacteria fight off viruses stumbled upon nature's own gene editing system. It's like we found the ultimate Swiss Army knife just lying there in the microbial world, waiting for us to figure out how to use it. By the end of this video, you'll understand why many scientists believe CRISPR represents the single most important technological breakthrough since the discovery of DNA itself. All right, let's start with the basics. What exactly is CRISPR, and how does this molecular miracle actually work? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. I know, that's a mouthful that tells you absolutely nothing. So let me give you a better explanation. Imagine your DNA as a massive library with 3.2 billion books, and each book contains the instructions for making different parts of you. Now imagine you had a librarian with supernatural abilities who could instantly locate any specific sentence in any specific book in that massive library, erase it, and write in a new sentence of your choosing. That librarian is CRISPR. But here's the incredible part. This technology didn't come from some high-tech laboratory, it came from bacteria. Yes, those tiny microbes that have been around for billions of years had already invented the ultimate gene editing system, and they were using it as their immune system. Here's how bacteria discovered genetic engineering. When a virus attacks a bacterium, the bacterium doesn't just fight it off. It takes a mugshot. It captures a piece of the virus's DNA and stores it in its own genome as a memory. These stored viral sequences are what we call CRISPR arrays. They're like a genetic most wanted poster. The next time that same virus shows up, the bacterium recognizes it immediately. It makes a copy of the stored viral DNA sequence and uses it as a guide to find and destroy the invading virus's genetic material. It's like having a molecular bouncer that never forgets a face. The key players in this system are two components, the guide RNA and the CA's protein. Think of the guide RNA as a GPS system that knows exactly where to go in the vast genome. It's a piece of RNA that matches perfectly with the target DNA sequence, like a key that fits only one specific lock. The CA's protein, usually Cas9, is the molecular scissors. It's the enforcer that actually does the cutting. When the guide RNA finds its target, Cas9 cuts both strands of the DNA at that exact location. It's like having a surgeon with perfect vision who never misses their target. But here's where it gets really clever. Once CRISPR cuts the DNA, the cell's natural repair machinery kicks in, and this is where we can choose what happens next. We can either let the cell repair itself naturally, which might disable a problematic gene, Or we can provide a new piece of DNA as a template, essentially giving the cell a blueprint for exactly how we want it to repair itself. Let me give you an analogy that really drives this home. Imagine you have a recipe book for making chocolate chip cookies, but there's a typo in the recipe that makes them taste terrible. Traditional medicine would be like giving you antacids after you eat the bad cookies, treating the symptoms but not fixing the problem. CRISPR is like having a molecular editor that can go directly to that recipe, erase the typo, and write in the correct ingredient. Now, every cookie you make from that recipe will be perfect. The precision of this system is absolutely mind-boggling. We're talking about making changes to individual letters in a genome that contains 3.2 billion letters. To put that in perspective, if the human genome were a book, it would be about 800,000 pages long. CRISPR can find and edit a single letter on a specific page in that book. That's like finding one specific grain of sand on a beach and painting it a different color. But the original CRISPR-Cas9 system was just the beginning. Scientists have now developed multiple generations of this technology, each more precise and powerful than the last. We have base editors that can change individual DNA letters without cutting the DNA at all, like using whiteout and a pen instead of scissors. We have prime editors that can insert, delete, or replace chunks of DNA with unprecedented accuracy. The latest advancement involves combining CRISPR with artificial intelligence. AI systems can now design guide RNAs that are more accurate, predict potential off-target effects, and even suggest the best editing strategies for specific genetic conditions. We're essentially giving CRISPR a brain to make it even smarter and more precise. And here's something that blew my mind when I first learned it. CRISPR components are incredibly small and stable. The guide RNA and Cas9 protein can be packaged into tiny delivery vehicles and sent anywhere in the body. They can cross the blood-brain barrier, target specific organs, and even be designed to activate only in certain types of cells. It's like having a molecular postal service that always delivers to the right address. Now that you understand how CRISPR works, let's talk about what it's doing right now to save lives and cure diseases that have been humanity's greatest enemies for millennia. The first thing you need to know is that we're not talking about future possibilities anymore. We're talking about patients who are walking around today completely cured of genetic diseases, thanks to CRISPR. Let's start with Victoria Gray. She was one of the first patients to receive CRISPR treatment for sickle cell disease. Victoria had spent her entire life in agony, frequent hospitalizations, pain crises that felt like her bones were being crushed from the inside, and the constant knowledge that her disease could kill her at any time. Victoria's treatment was revolutionary. Doctors extracted her bone marrow cells, used CRISPR to edit them so they would produce healthy hemoglobin, and then reinfused them back into her body. Within months, her body was producing normal, healthy red blood cells for the first time in her life. She hasn't had a pain crisis since the treatment. She's essentially been cured of a disease that was supposed to kill her. But Victoria isn't alone. The CRISPR treatment called Casgevy has now been approved in the United States, United Kingdom, and other countries. Clinical trials have shown success rates that are almost unbelievable. Over 95% of patients become transfusion independent, meaning they no longer need regular blood transfusions to survive, and that's just sickle cell disease. Let's talk about beta thalassemia, another blood disorder where patients require blood transfusions every few weeks just to stay alive. CRISPR treatments for beta thalassemia have shown similar miraculous results. Patients who have been dependent on transfusions their entire lives are now producing their own healthy blood cells. But blood disorders are just the beginning. CRISPR trials are currently underway for dozens of other conditions, and the results are consistently mind-blowing. Take Leber congenital amaurosis, a genetic form of blindness that affects children. Researchers have used CRISPR to restore vision to patients who have been blind since birth. Cancer treatment is another area where CRISPR is showing incredible promise. CAR T-cell therapy using CRISPR-edited immune cells has achieved complete remission in patients with blood cancers that were completely resistant to all other treatments. The process involves taking a patient's T-cells, the soldiers of the immune system, editing them with CRISPR to make them better at recognizing and destroying cancer cells, and then putting them back into the patient. But here's where it gets really exciting. Researchers are working on off-the-shelf CAR T-cells. Instead of using each patient's own cells, they're creating universal donor cells that have been edited with CRISPR to be compatible with any patient. This could make cutting-edge cancer treatment available immediately, without the weeks of waiting required for personalized treatments. Duchenne muscular dystrophy is a devastating disease where children progressively lose muscle function and typically die in their teens or 20s. CRISPR gene therapy trials have shown that they can restore the production of dystrophin, the protein that's missing in these patients. Early results show not just halting the progression of the disease, but actually reversing some of the muscle damage. HIV treatment is another breakthrough area. Scientists have used CRISPR to edit the CCR5 gene in patients' immune cells, essentially making them resistant to HIV infection. Some patients who received this treatment have been able to stop taking antiretroviral drugs while maintaining undetectable viral loads. Inherited forms of blindness, deafness, heart disease, liver disease, lung disease. There are currently over 40 different CRISPR clinical trials running for genetic diseases. And the success rates are consistently higher than anyone expected. But perhaps the most exciting development is the emergence of in vivo CRISPR treatments, therapies that are delivered directly into the body without removing cells first. These treatments use specially designed delivery systems to get CRISPR components to the right tissues. Base editing, one of the newer CRISPR technologies, is showing particular promise for treating diseases caused by single-letter changes in DNA. Instead of cutting the DNA, base editors can directly convert one DNA letter to another. This is perfect for diseases like sickle cell, where the only problem is a single A that should be a T. What's particularly remarkable is how fast these treatments are moving from lab to clinic. Traditional drug development takes 15-20 years from discovery to approval. CRISPR treatments are going from concept to clinical trials in just a few years. The first CRISPR medicine was approved just 11 years after the technology was first described in scientific papers. The patients receiving these treatments aren't just getting better, they're being completely transformed. Parents are watching children with terminal genetic diseases grow up healthy. Adults who were facing certain death are planning for decades of healthy life. We're witnessing the end of genetic disease as an inevitable human tragedy. While CRISPR's medical applications are grabbing headlines, there's another revolution happening that could be even more transformative for humanity's future. CRISPR's applications in agriculture, environmental protection, and species conservation. We're not just talking about treating diseases, we're talking about feeding the world, saving the planet, and potentially bringing back species that have been extinct for thousands of years. Let's start with food security because this might be the most important challenge facing humanity in the next few decades. By 2050, we'll need to feed nearly 10 billion people on a planet where climate change is making traditional farming increasingly difficult. CRISPR might be our secret weapon. Traditional crop breeding takes decades to develop new varieties. With CRISPR, we can make precise improvements to crops in just a few years. And I'm not talking about minor tweaks. I'm talking about game-changing improvements that could revolutionize agriculture. But it goes beyond just drought resistance. Scientists have created CRISPR-edited crops that are resistant to devastating plant diseases that currently destroy millions of tons of food every year. Rice blast disease alone destroys enough rice annually to feed 60 million people. CRISPR-edited rice varieties are completely resistant to this disease. And here's something that really blew my mind. Scientists are using CRISPR to improve the nutritional content of staple crops. Golden rice enhanced with CRISPR to produce high levels of vitamin A could prevent blindness and death from vitamin A deficiency in millions of children worldwide. One of the most exciting developments is the creation of crops that can actually help combat climate change. Scientists have edited plants to have deeper root systems that can capture and store more carbon in the soil. Some estimates suggest that if these carbon-capturing crops were widely adopted, they could sequester enough CO2 to significantly slow climate change. But CRISPR's environmental applications extend far beyond agriculture. Researchers are using gene editing to address some of our most pressing environmental challenges. Take the fight against invasive species. Invasive plants and animals cause billions of dollars in damage every year and threaten native ecosystems worldwide. The most ambitious application might be in coral reef conservation. Coral bleaching caused by rising ocean temperatures is killing reefs worldwide. Researchers are using CRISPR to edit corals to make them more resistant to heat stress and ocean acidification. Early trials have shown that CRISPR-edited corals can survive temperature increases that would kill normal corals. And then there's the mind-blowing possibility of de-extinction, bringing back species that have disappeared from Earth. Scientists are using CRISPR to edit the genomes of closely related living species, gradually converting them into genetic approximations of extinct species. The woolly mammoth project is the most famous example. Researchers are editing elephant cells to incorporate mammoth-specific genes for cold tolerance, fur production, and other mammoth characteristics. The goal isn't just scientific curiosity. Woolly mammoths could potentially help restore grassland ecosystems in the Arctic, which could help slow climate change by preventing permafrost from melting. CRISPR is also being used for environmental cleanup. Researchers have edited microorganisms to break down plastic pollution, clean up oil spills, and neutralize toxic compounds in contaminated soil and water. These biological cleanup systems could be much more effective and environmentally friendly than current chemical approaches. The scale of what's possible is almost hard to comprehend. We're talking about reshaping entire ecosystems, bringing back lost biodiversity, creating food security for billions of people, and potentially reversing some of the damage that human activity has done to the planet. Now let's talk about the future. And this is where things get really wild. Because while everything we've discussed so far is incredible, it's nothing compared to what's coming next. We're standing at the threshold of capabilities that will fundamentally change what it means to be human. The next frontier for CRISPR isn't just treating disease, it's enhancing human capabilities. And I'm not talking about science fiction scenarios. I'm talking about technologies that are currently in development and could be available within the next decade. Enhanced intelligence is particularly intriguing. Scientists have identified genes that influence brain development, memory formation, and cognitive function. In animal studies, editing these genes has produced dramatic improvements in learning ability and memory. But here's where things get really mind-bending. Germline editing. This is CRISPR modification of sperm, eggs, or early embryos that would be passed on to future generations. This technology could potentially eliminate genetic diseases from human bloodlines forever. The technical capabilities for germline editing already exist. In 2018, a Chinese scientist named He Jiankui used CRISPR to edit human embryos, creating the first gene-edited babies. While his work was widely condemned for ethical violations, it proved that the technology works. But with great power comes great responsibility, and CRISPR raises profound ethical questions that humanity has never had to grapple with before. The first major concern is safety. While CRISPR is incredibly precise, it's not perfect. Then there's the question of enhancement versus treatment. Most people agree that using CRISPR to cure genetic diseases is ethical. But what about using it to make people stronger, smarter, or more attractive? Where do we draw the line between medical treatment and human enhancement? This leads to concerns about creating a genetic divide in society. If CRISPR enhancements are expensive, we could end up with a world where genetic advantages are only available to the wealthy, creating a biological class system that's even more fundamental than current economic inequalities.